Hello students, myself Shazeen Khan from Department of Botany, and we are here to discuss the topic of regulation of Kelvin cycle for MSc second year, credit 2 from the subject of plant physiology. In the last lecture, we had seen the various steps of Kelvin cycle in detail. We all know that Kelvin cycle, it is the major photosynthetic cycle which is used to fix CO2 in the presence of sunlight, and it is the main process of photosynthesis which fixes carbon dioxide to prepare food for the plants. All that we have seen in detail in the last lecture. In today's lecture, we will be seeing how the Kelvin cycle is regulated. We will see the different mechanisms, how the different enzymes in the Kelvin cycle, they are regulated. So let us start with the introduction. We all know that the Kelvin cycle, also known as the C3 cycle, it was discovered by Kelvin, Benson and their colleagues using unicellular algae chlorella. We all know that it is a light-independent reaction which takes place in the stroma of the chloroplast, and it consists of different phases, namely carboxylation phase, the reduction phase, synthesis phase, and the regeneration phase. And we also know that why it is known as C3 cycle, because the first stable product of this cycle, it is a 3-carbon compound, which is 3-phosphoglyceric acid. So that is why this cycle, it is known as C3 cycle. So we will be seeing that why this cycle, it has to be regulated. The high efficiency of Kelvin cycle, it indicates that there is some form of regulation present, and it ensures that all the intermediates in the cycle, they are present at adequate concentrations, and that the cycle, it is turned off when it is not needed. So we will be seeing that when and how this cycle turns on and turns off. It should be turned off when it is not in use, so that the resources, they are not wasted. So we will see different enzymes first, that which are the enzymes in the C3 cycle which are regulated for the regulation of the overall cycle. So there are 5 enzymes which you can see on the screen here. First is Rubisco, which is your ribulose 1,5-bisphosphate carboxylase, the major enzyme of the Kelvin cycle. Second enzyme is the NADP glyceraldehyde 3-phosphate dehydrogenase. Third, we have fructose 1,6-bisphosphatase. The fourth enzyme is pseudoheptulose 1,7-bisphosphatase, and the last enzyme is ribulose 5-phosphate kinase. First, we will see that which reactions do these enzymes catalyze. So the first and the major enzyme of the C3 cycle we will see first, and we all know that it catalyzes the reaction between ribulose 1,5-bisphosphate and carbon dioxide to form the first stable product of the Kelvin cycle, that is 3-phosphoglyceric acid. The second enzyme is NADP glyceraldehyde 3-phosphate. It catalyzes the reaction between 1,3-bisphosphoglyceric acid and NADPH to form 3-phosphoglyceraldehyde. Let us see the next enzymes. The next 2 enzymes, we have fructose 1,6-bisphosphatase. It catalyzes the reaction to form fructose 6-phosphate from fructose 1,6-bisphosphate. Similarly, we have pseudoheptulose 1,7-bisphosphatase. It catalyzes the reaction to produce pseudoheptulose 7-phosphate from pseudoheptulose 1,7-bisphosphate. In these two enzymes, we all know that it removes one phosphate group from the reactant and it forms the product. So both these enzymes, they have somewhat similar function. And the last enzyme is ribulose 5-phosphate kinase. This enzyme, it catalyzes the reaction to produce ribulose 1,5-bisphosphate from ribulose 5-phosphate, that is your RUBP compound, which is the first first step in the Kelvin cycle. So this enzyme, it is used in the regeneration step, that is for the regeneration of the reactants in the Kelvin cycle. So these are the 5 enzymes which we have seen, the major enzymes whose regulation we will study to study the overall regulation of Kelvin cycle. So from these 5 enzymes, the first enzyme Rubisco, it has some different mechanisms of regulation, and the other 4 enzymes, they are regulated by the Ferredoxin thioredoxin system. So we will see each of the regulation one by one. First, we will see the regulation of Rubisco enzyme. This enzyme, it is inactive until carbamylated on the amino group of lysine amino acid. Now you all know that enzymes, they have a certain active site present on them. On this active site of Rubisco, an amino acid lysine is present, which you can see on your screen, lysine 201. Now this active site, what happens is a compound, that is ribulose 1,5-bisphosphate, it binds tightly on the active site of this enzyme. So when this compound, it binds tightly on the active site, it makes the enzyme Rubisco inactive. So how this inhibition is overcome? There is another enzyme, that is Rubisco activase. It releases that ribulose 1,5-bisphosphate, and then the enzyme Rubisco, it becomes active. We will see that in the next slide. You can see the diagrammatic representation here. You can see the enzyme that is blue in color. Rubisco is written on the enzyme. And on the active side, you can see an amino acid lysine present. Now the amino acid lysine, it is present on the active site. And that active site, it gets covered because of a compound present there. You can see the compound there, ribulose 1,5-bisphosphate. It is binding tightly on the active site and it is covering the active site. So how this inhibition is overcome, that by another enzyme, that is Rubisco activase. What it does, it releases that ribulose 1,5-bisphosphate with the help of ATP. It is ATP dependent release. And when this ribulose 1,5-bisphosphate is released from the active site of the enzyme, a molecule of carbon dioxide comes and binds on the amino group of lysine. Also, magnesium ion acts as a cofactor and it forms a complex there. So this complex, as a result, activates the enzyme Rubisco. So in this way, this is one of the mechanism of activation of the enzyme Rubisco. Another mechanism is also there for the activation of Rubisco. What happens during the dark time? There is one inhibitor. It is a nocturnal inhibitor. Why? Because it inhibits the enzyme during dark. And which is this inhibitor? It is 2-carboxy-arabinitol 1-phosphate. So this molecule also, it binds on the active site of Rubisco enzyme. Even plain Rubisco and carbomylated Rubisco also. Carbomylated Rubisco we have seen in the last slide. You can see here at the bottom, carbomylated Rubisco. Carbomylated Rubisco means the binding of carbon dioxide and magnesium ions. That is the carbomylated Rubisco. So during dark time, this compound, that is 2-carboxy-arabinitol 1-phosphate, it binds on the carbomylated Rubisco. And how it is released? It is released with the help of sunlight. It is broken down when light returns or it is expelled by Rubisco activase. So this is a nocturnal inhibitor, that is, it inhibits the enzyme during dark time or during night time. So this is also one more mechanism of activation of Rubisco enzyme. Now the other 4 enzymes which were written in the list, those are activated by some other mechanism, and that is the Ferredoxin thioredoxin system. The other 4 enzymes which are present in the list, those all 4 enzymes, they are activated by Ferredoxin thioredoxin system. So this is a covalent thiol-based oxidation reduction mechanism. These enzymes are activated by light-driven reduction of disulfide bonds between 2 cysteine residues critical to their catalytic activities. What happens is, enzymes, they have some cysteine residues present in them. So when these cysteine residues, they are present in oxidized stage, that is a disulfide stage, it makes these enzymes inactive. And this inactive stage, it is a very normal condition during dark time. That is, during night time, these enzymes, they are present in the disulfide stage or in the oxidized stage. During daytime, this disulfide stage or the oxidized stage, it is reduced to form sulfahydryl stage, and this stage, it is the activated form of enzyme. So how this activation happens, let us see this. You can see in the diagram how this mechanism takes place. What happens is, during daytime in photosystem 1, we have the cyclic photophosphorylation. So that happens, that is a light dependent phase. So during daytime, when photosystem 1, it absorbs light, the ferredoxin, which is in the oxidized stage, it becomes reduced. And this reduced ferredoxin then passes on the electrons to the thioredoxin, and oxidized thioredoxin, it becomes in the reduced form. And all these processes, it takes place because of one enzyme you can see there in the diagram. That enzyme is ferredoxin thioredoxin reductase. So you can see how the electrons, they are transferred from an oxidized stage to reduced stage, and the oxidized stage, it is getting reduced to become an active enzyme. The oxidized stage, it is disulfide state, and the reduced stage, it is the sulfahydryl stage. So you can see ferredoxin, it was in the oxidized stage. When light was present, it got reduced to active form. This reduced ferredoxin then transfers electrons to oxidized thioredoxin. This oxidized thioredoxin, with the help of ferredoxin thioredoxin reductase, it forms reduced thioredoxin, and this activated reduced thioredoxin then targets the different enzymes of the Calvin cycle which we have seen, and those which were in inactive form, they get activated. And how they are activated? Because the disulfide group, it is reduced to sulfahydryl stage. So remember that whenever the reduced form of enzymes are present, they are activated, or they are active form of the enzymes. So in this way, the different other 4 enzymes, they are activated by the ferredoxin thioredoxin system. Now some other mechanisms are also present which regulate the different Calvin cycle enzymes, and those are light dependent ion movements. What are these mechanisms? Let us see them also. Now light causes several ion changes which are reversible ion changes in the stroma of the chloroplast, and these changes, it influences the activity of Rubisco and other chloroplast enzymes. What happens is, during daytime upon illumination, protons or the hydrogen ions, they are pumped from the stroma into the lumen of thylakoids. So when these protons are transferred from the stroma to the lumen, magnesium ions are taken, or there is an uptake of magnesium ions in the stroma. So what does these ion fluxes result in? That is, you can see that protons are pumped out of the stroma. So if protons concentration is decreasing, what happens is the pH of stroma, or the stromal concentration, stromal pH, it increases. You can see that pH 7 to 8, there is increase of pH. And also there is increase in the concentration of magnesium ions. So due to this proton efflux and magnesium ion influx, there are different changes which occur in the stroma. And these enzymes, different enzymes of the Calvin cycle which we have seen, they are more active at a pH 8 rather than pH 7. And we have seen that because of sunlight, these protons, they are pumped out. So the pH has increased from a slightly acidic pH, it has gone to a basic or alkaline pH. So pH 8, these enzymes are more active. And we also know that magnesium ion, it acts as a cofactor for the catalysis or for the working of these enzymes. So this is one, what you call, mechanism, that light dependent ion changes also, it activates these enzymes. Now these changes in the ionic composition of chloroplast stroma, they are reversed upon darkening. So all these mechanisms which we are seeing, they are usually happening in the daytime. During night time, all these processes, they are reversed, and these enzymes will get inhibited. So to conclude this session, we saw the different mechanisms of the Calvin cycle, how they are regulated. And this regulation is important as it activates the enzyme only when they are required, or else the functioning of the cycle is turned off. Also, the regulation of the cycle ensures that there is no futile cycling of products and the Kelvin cycle operates only when it is needed. We have seen different mechanisms today, and we can conclude that the different enzymes, they are inhibited during dark time. Why during dark time? Because photosynthesis, it is light dependent. It takes place only during the daytime. So during dark time, this cycle, or during night time actually, this cycle, it should not take place. There should be no wasting of resources. So these enzymes, they are inhibited during the dark, and what you call wasteful cycling or futile cycling of products, it is inhibited or it is prevented. So that is why this regulation of Kelvin cycle is important, so that the cycle is turned on only when it is needed. So this is the student assignment for you. You will write in brief about Kelvin cycle, different steps, and you will mail it on the given email ID. The summary of today's session, if we see, we have seen the different enzymes of Kelvin cycle which are useful in the regulation of the Kelvin cycle. We have seen 5 enzymes: Rubisco, NADP glyceraldehyde phosphate, fructose 1,6 bisphosphatase, pseudoheptulose 1,7 bisphosphatase, and ribulose kinase. So these 5 enzymes we have seen. The first enzyme, it is Rubisco. It is activated by the ATP dependent release of the inhibitors. We have also seen that there was a nocturnal inhibitor present in this enzyme, and we have seen that different ionic changes also activate this Rubisco enzyme. The other 4 enzymes in the list, those enzymes, they are activated by the Ferredoxin thioredoxin system. And we have seen the overall regulation of Kelvin cycle and why it is important. In the next lecture, we will be talking about photorespiration, which is also known as the C2 cycle. We will be seeing the different steps and the mechanism of photorespiration, the significance of this cycle. Thank you so much for your time. Thank you for watching today's lecture.